Hi everyone, welcome to our second week of classes. This week we're going to change gears a bit and focus a bit more on structure, on anatomy, and that behooves us because after all this is a neuroscience course. So before we proceed with a detailed discussion of function, it is highly beneficial to understand how the brain is structured and what are the building basic blocks of neural communication, of information transmission within the brain. Accordingly, the lecture is structured in two parts. First, we're gonna review and highlight some of the important aspects of neuronal function and neurotransmission. And I say review because I assume that prior knowledge exists on these topics from Intro to Psych. And if not, then the textbook also provides a very good description of this topic. Secondly, we're gonna be discussing macro anatomy, discussion of brain structure at a larger level with focus on different key areas in cognition and in brain function. And we'll be paying a bit more attention to this topic, primarily because it brings us closer to some of the topics that we will have the chance to discuss in detail this term. Okay, so quickly going through the objectives of the lecture today. First, we want to understand how neurons are able to send signals using both electrical and chemical transmission. It's hard to fully appreciate what's going on in the human brain without having some understanding of neurotransmission. However, to appreciate the practical relevance and the clinical relevance of good, normal neurotransmission, we'll also look at what happens in some disorders and diseases, such as multiple sclerosis and amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, as well as a stroke. Further, we'll try to identify the major pathways of the spinal cord in the central nervous system. And then we'll move on to identify some of the key players in terms of cortical regions and also a couple of subcortical areas in terms of cognitive functioning. We will try to gain a basic understanding of how to navigate the human brain, for instance, across what directions, and also apply this knowledge whenever we're discussing the relationship of different brain regions with each other from an anatomical standpoint. And last but not least, we aim to identify some of the basic mechanisms that play a neuroprotective role. So with that being said, let's get started. And while much of the focus in neuroscience is on neurons and neural transmissions, we are going to start our discussion with glial cells. And this is because primarily we want to do due diligence and we want to make them justice. Non-neural cells or glial cells are some of the most misunderstood aspects or components of the neural system. And they play a fundamental role in the functioning of the healthy brain. So where does this misunderstanding come from? Well, what are glial cells first? They're non-neural cells. Unlike neurons, they do not generate electrical impulses. However, because of that, it was thought that they play no role in neurotransmission, and that appears to be wrong. And clarifying their role in neurotransmission nowadays is a very active field of research. But perhaps an even more conspicuous misunderstanding of glial cells is in terms of number. glial cells take their name from the initial hypothesis that they provide the glue of the nervous system. They provide a scaffolding for the human brain. And that is not false. That is largely correct. However, associated with that was the belief that the number of glial cells is in a ratio of 10 to 1 relative to neurons, and that appears to be quite false. In fact, the number of glial cells is roughly in a 1-to-1 proportion with the number of neurons in the healthy adult human brain. And the other one was that they're rather unilateral in terms of structure and function. And here we couldn't have been more wrong, because glial cells appear to exhibit quite a variety in terms of cellular structure, and also related to that in terms of function. And we don't have a lot of time to go into the full range and diversity of structural and functional roles that the glial cells perform. However, we will quickly look at 4 different types of glial cells and their corresponding function. And perhaps the best well-known type of glial cell is the astrocyte, which takes its name from being star-shaped. And what most neuroscientists know astrocytes for is the fact that it is responsible for providing the blood-brain barrier. That is, the astrocytes form a protective barrier along blood vessels and allow only some things to pass from the blood into the nervous tissue. For instance, this blood-brain barrier allows hormones and oxygen, which is now is fundamental for the metabolic costs of neural functioning in the brain, into the neural tissue. However, it prevents some pathogens and other chemical compounds, for instance, a variety of bacteria, for passing from blood into the rest of the brain. And this is fundamental for protecting the integrity of brain and its function. It is also nowadays apparent that the astrocytes are far more diverse in terms of what they can and do accomplish. So, for instance, it is currently known that astrocytes are quite important in neurodevelopment. So they provide the initial scaffolding of the developing brain. And also it is known that they play a role in neural repair. And again, these are very active fields of research nowadays. Okay, so what other types of glial cells are there? And 2 that are particularly relevant for neural transmission, as we'll have the opportunity to see in a couple of minutes, are oligodendrocytes and Schwann cells. And they more or less perform the same function. It's just the case that oligodendrocytes reside in the central nervous system, which, as you might remember, is comprised of the brain and the spinal cord, while Schwann cells reside in the peripheral nervous system. So what is this function? Well, simply put, they provide myelination. Myelin is a lipid layer, fatty layer surrounding the axon, which plays a key role in neural transmission, in providing quick and effective neural transmission. And we'll delve into that topic just in a few minutes. Okay, and one other type of glial cell we should mention is the microglial cell. This is a rather irregular shape, and it is also known to reproduce quite in high numbers, even in the healthy adult brain, unlike other non-neural cells or unlike neurons. But more importantly, is the function that microglial cells perform, and that is scavenging. They essentially take care, they consume dying or dead neural cells, and this is quite important for the health of the well-functioning brain. Okay, so glial cells very broadly cover a wide spectrum of functions and quite a few different types associated with rather different cellular structures. They are perhaps the most misunderstood up to this day in terms of their function and structure. Nevertheless, at this point, we do have to move on. And much of the focus in neuroscience, including cognitive neuroscience, is on neurons and neurotransmissions. So we're going to delve into that next. It should come as no surprise that just as we have an entire variety of types of glial cells, we also have a variety of neural cells. However, broadly, we can categorize them in three groups: motor neurons, sensory neurons, and interneurons. The first group, motor neurons, unsurprisingly, based on their name, reside in the motor cortex, the brainstem, and the spinal cord, and they're responsible for connecting with muscles and glands. They're ultimately responsible for motor control, for movement. Sensory neurons reside at the other end of this input-output mechanism, which is provided by the nervous system. They're responsible for receiving information, and according to the sensory modality, we can have very different types of neurons. So, for instance, we have photoreceptors on the retina, which are responsible for converting light into neural signals. We have auditory receptors that convert pressure waves, air waves, into neural signals. We have odor receptors that are sensitive to odor molecules. And we also have things such as nociceptors, neurons that are responsible to damage being done to the outward of the body, right? So let's say to a change in temperature, which could be potentially damaging to us. Or on the surface of internal organs. So such sensory neurons are not necessarily just external, but also internal. They keep track of what's going on outside of our body and inside of our body. So nociceptors, pain receptors, are responsible for keeping track of damaging things happening on the outside or outside. Pain, whether external or internal. Equally important, though, is another group of neurons, which constitute about 20 to 30% of the entire population of neurons in the CNS, in the central nervous system. And interneurons serve to relay information, to relay signals between sensory neurons and motor neurons. But perhaps equally important, also to integrate information. We can have entire groups of interneurons receiving information from sensory neurons, and then aggregating that information with the aim of distilling it into something more relevant, right? It can emphasize information and can filter out information. It can sum up information, aggregate it in a variety of ways. And because of that, interneurons are key to learning and decision making. We learn how to mediate the relationship between the sensory input and the motor output by enabling these interneurons to do different things in a different manner. So they're fundamental to neural functioning and neural information processing, right? If you think about signals as the building blocks of information processing by the brain. So it goes without saying that because all these different types of neurons serve different purposes, there is a large variability in terms of structural architecture, right? So for instance, you might imagine that a sensory neuron will have lots of branching for its dendrites through which it accumulates, it aggregates informations from tens or hundreds of thousands of other neurons. While other categories of neurons, perhaps some interneurons, which are less relevant in that sense, might not have such an extensive branching of their dendrites. So structure is very often related to function, and we'll have the opportunity to revisit this idea at different levels of the anatomical lens, and also with respect to different parts of the brain. But some things remain common across all this diversity. So perhaps it's worth spending a few minutes just looking at the structure and function of a standard typical neuron, as we often conceptualize it and describe it in introductory classes. And you should already be somewhat familiar with this. So this is only a very brief and cursory review of the fundamental structure of a neuron. But what you might already remember is the fact that neurons stand out from other cells by the fact that in addition to a cell body or soma, they also exhibit what's called dendrites, and we already mentioned them, through which they receive input, they receive and integrate signals from other neurons, let's say, as well as an axon, which is responsible for transmitting the neural signal down to other neurons through axon terminals. So informally, you can think of dendrites as an antenna, as a neural antenna, a way for receiving information, and you can think of the axon for the pathway along which that information is transmitted to other neurons. Or in the case of motor neurons to muscles and glands. Okay, so now this is actually quite a complex architecture, and there's a lot of variability around it. I already mentioned the fact that dendrites, all this branching extension of the cell body can be more or less accentuated, and that relates to function. Also, the length of the axon is quite variable. So again, it depends on what function the actual neuron subserves. So if, let's say, we're dealing with an interneuron whose function is to relay information, then probably the axon will be a lot longer, right? While if the function of the interneuron is primarily to communicate with a patch of near neighbors, other interneurons, in the service of integrating information, then the axon is probably a lot shorter. So lots of variability. Still, if we think about a typical neuron, this is by and large its structure. So then an important question is, how does this structure subserve neural communication, neural transmission? And a fundamental aspect of that process of neural transmission is the membrane potential. So everything seems to happen because the neuron is primed, so to speak, to communicate, to generate an electrical impulse. So what does this mean? What is this membrane potential? Well, this happens because a certain balance that takes into account the concentration of ions on the outside and on the inside of the cell, of the soma, as well as the relative difference in voltage associated with these ions. So there is quite a complex mechanism which is responsible for keeping this balance in place. And as a result of this balance, what happens is that the inside of the membrane is negatively charged with respect to the outside. There is larger concentration of negative versus positively charged ions within the cell. And the way in which this is important is because the membrane potential, which is roughly of minus 70 millivolts, is functional in providing a quick mechanism for neural transmission. But before we go into that, let's take a cursory look at how that negative potential is maintained. And this all has to do with the semi-permeability of the membrane surrounding the body of the neuron, the soma, and the presence of a variety of channels that allow the restricted passage of ions in and out of the cell. Okay, so now there is a variety of types of channels, but perhaps one of the fundamental distinctions here is between gated and non-gated channels. So non-gated channels are responsible selectively for allowing the passage in and out of, let's say, sodium or potassium. But one of the main mechanisms for maintaining the membrane potential is provided by the gated ion channels. So what these gated channels do, especially what we call pumps, are to push out of the cell sodium ions at the cost of bringing in potassium ions. So you may think this is an equal trade. Positive ions go out and positively charged ions go in. However, at the same time with these pumps that work quite hard, and they are some of the main consumers of energy associated with the metabolic cost of keeping neural processes running. And we're going to go back to that topic a bit later. But in addition to them, we also have the non-gated channels that serve to eliminate a lot of the potassium ions. So because of this interesting mechanism that balances electrical constraints and chemical constraints, chemical concentration, we end up with a negatively charged inside membrane potential. Okay, so this is very briefly what happens at the level of membrane, and this is how this membrane potential arises typically. So then how do we move on from this resting membrane potential to an action potential, to the generation and transmission of an electrical signal? Well, this has to do first with the accumulation of signals from the dendrite and their communication, the passage of these signals onto the soma. So we'll cover the topic of dendrite functioning in a second, but for simplicity, let's just say that all those signals are passed onto the soma and then onto the axon hillock. And then what happens is that once sufficient depolarization happens, then as soon as a certain threshold is passed, that depolarization is further amplified to generate an action potential, a considerable further depolarization of the inside of the cell, of the membrane. And there are key aspects of this process that you should pay attention to and you should be aware of. First of all, it doesn't really matter how much depolarization happens. in terms of amplitude. The only thing that really matters is whether that depolarization is triggered by what happens at the level of the dendrites, how much signal is accumulated at the level of the dendrites and passed on to the soma. The only thing that matters is whether that depolarization passes or doesn't pass the threshold. If it doesn't pass that specific threshold, then the resting membrane potential is reestablished quite rapidly. However, if that depolarization passes a threshold, then this considerable spike in depolarization results as an amplification on the original depolarization. And this is an interesting trait. The action potential does not vary in amplitude. It's an all or none signal, so it either happens or it doesn't happen. And if it does happen, it always registers more or less the same amplitude. So then a quick key question here, and a quite normal one at this stage, is how are we sensitive, or rather how is an individual neuron sensitive to the amount and intensity of the input that it receives? And this happens not through the amplitude of an action potential, but rather through the frequency with which an action potential happens. And there is a physiological limit to how often action potentials can occur. However, there is a great variability still in terms of possible frequency. Also, we refer to action potentials as spikes because this is often how they are seen whenever you record their activity through intracellular electrodes. Okay, so this is how an action potential is triggered. And it is a fairly short, very brief spike, yet the significant one. And once a spike is registered, then the signal relatively quickly is restored to the membrane potential after passing through a short overpolarization phase. And once that happens, then we may expect the occurrence of another action potential. Okay, so all of this rather complex mechanism and its outcome resides primarily at the location of the axon hillock. So the very beginning of the axon. So then how is this signal transmitted all the way throughout the axons? And keep in mind that the length of the axon is quite variable, and some of these axons are quite long, perhaps not as long in humans, but in other animals they can be meters long. Well, one problem with simply passing this information in one step from the axon hillock all the way to the axon terminals is the fact that there is a decrement with which the level of electrical depolarization occurs. So the further we go from the axon hillock, where the action potential was generated, the more we run the risk of losing that depolarization. And the clever mechanism that is used by our biology to help us preserve the integrity of that signal is through myelination. So you might already remember that some glial cells are responsible for embedding, or rather encapsulating, the axons in a myelin sheath, which serves as an electrical insulator. However, this is not a continuous sheath. It's rather a series of patches interrupted by uninsulated portions of the axon membrane. And these are the nodes of Ranvier, which stand out by the fact that not only are they not insulated, but they have a high concentration of ion channels. So what happens is that as soon as that depolarization, as soon as the action potential reaches one of these nodes of run via, then the ion channels serve to reinforce and reestablish the strength of that action potential. And then we can move on step by step from one node to the next until we reach the axon terminal at no real cost, at no significant cost in the strength or the ability to preserve the action potential. And because of this, we call the process of passing information, of passing the action potential across the axon, saltatory conduction, from the word saltare, to jump in Latin. So this is briefly how the action potential is propagated down the axon. Now, one way to practically and intuitively appreciate the importance of normal functioning at the level of the neurons and neural transmission is to see what happens if these things go wrong. And perhaps one of the clearest demonstrations of what happens if neurotransmission is impaired, if action potentials do not serve their purpose effectively, is MS, multiple sclerosis. This is believed to be largely an autoimmune disorder. It is the result of the immune system being overactive, and as a result of that, progressively damaging the myelin that surrounds the axons. As a result of this, and as a result of where this takes place, patients can exhibit a variety of symptoms. So for instance, if the condition attacks motor fibers, that you might expect problems with movement. If the condition attacks the optic nerve, you expect problems with vision. If it attacks the neurons of the temporal lobe, then you expect memory problems. And this variety is problematic when it comes to diagnosing the disease. So you might imagine, for instance, if this happens in an older adult, we might be inclined to diagnose it as Alzheimer's before further tests. And it does take a variety of lab tests and scans to confirm the nature of the disease. To confirm that this is multiple sclerosis and not Alzheimer's. So this is believed to be the origin, the autoimmune source of the disease, and these are the symptoms. So right now, what about the causes? And unfortunately, we don't really know so much about it. It is believed that a variety of factors might contribute to it, such as climate, food, and so on and so forth. And this is a very interesting public health topic, especially as Canada sees one of the highest rates of multiple sclerosis in the world. What makes this situation even worse is the fact that presently there is no cure for MS, and available treatments emphasize primarily the symptoms, so managing symptoms and altering the course of the disease. If you try to acquire more information about this, and statistically speaking, it is likely that most of us know someone who suffers from MS. So all the more so, if you do try to volunteer in any way and help your community, or just find out more about this type of research, I would point out this link at the bottom, which links us to the MS Society of Canada to gather more information. Okay, moving on to another condition. This is the amyotrophic lateral sclerosis, or ALS, more commonly known as motor neuron disease or Lou Gehrig's disease. And this is a terrible condition that attacks upper and lower motor neurons, and ultimately results in death. It is a terrible condition because it attacks these neurons extremely fast for a neurodegenerative disease. So from the time at which you exhibit symptoms until the time of death, usually there is no more than 3 to 5 years. However, in some uncharacteristic cases, such as that of Stephen Hawking more recently, people can live much longer. Now, because this condition attacks the motor neurons, you might expect that the ability to control muscles is impaired over time, and this can also result in muscle atrophy. So eventually the outcome in terms of symptoms is inability, or rather first impaired ability to walk, to use your hands, but then also to swallow and to breathe. Why does this happen? There is believed to be a genetic component to this disease, but ultimately we don't really have a good understanding of its cause. Also, it is difficult to diagnose. So for instance, it can be easily confusable with some versions of MS. Hence, a lot of work is dedicating nowadays both in developing better diagnosis tools and treatment tools, especially since it is a deathly disease with no cure. What is the mechanism through which happens? This is somewhat better known, and it is attributed to excitotoxicity, to over-excitatory profiles of these neurons, of the motor neurons. And the few treatments available on the market attempt to down-regulate this excitotoxicity, and in doing so, not cure but slow down the symptoms and the course of disease. Okay, one other condition that we should talk about, both due to its prevalence and also to its relevance for neuropsychology, is stroke. And in particular ischemic stroke. And the reason for this is because it is one of the top 3 killers in this industrialized world, including Canada, and it is the leading cause of long-term disability. Now, what is the mechanism of the stroke? Oh, well, it's all got to do with the blood supply to the brain. There are 2 major types of stroke. There is an ischemic stroke, which is by far the most frequent one, and that involves a stoppage of blood flow to the brain due to a blood clot or a building plaque inside a vein. Now, if this happens, then an entire part of the brain may be depleted of blood and henceforth of oxygen, incapable of satisfying the basic metabolic costs for neural functioning and neurotransmission. So if this happens, one possibility is that what we see is electrical failure. So due to the reduced metabolic availability of resources, of energy, neurons are no longer able to transmit action potentials, or in worse circumstance, we see neuronal death. Okay, so this can happen because of ischemia. The other possibility is hemorrhagic stroke, which is the result of the blood vessel becoming ruptured or leaking. In any case, this is something that should be treated right away. The best outcome is where the treatment is provided within 2 or 3 hours. And there are certain symptoms that are quite well associated and able to distinguish between stroke and other conditions. So for instance, because the problem tends to impact one part of the brain quite rapidly rather than the other one, what a patient might experience is face drooping on one side of the face. So you can often think about it or associate it with an uneven smile. You can sense arm weakness or leg weakness associated with that, with the corresponding part of the body. It is also often associated with slurred speech or speech difficulty. But ultimately, the precise symptoms and the outcome of this condition differs a lot based on what part of the brain is impacted. And in many cases, this impacts more areas, but just like MS can be associated with a variety of symptoms depending on what part of the brain is associated, the same can happen with stroke. So for instance, we can also see memory problems happening if the medial temporal lobe is the area targeted. And if you study vision like I do and our TAs, then you can see problems with visual processing or high-level visual recognition if the occipital cortex or some parts of the IT of the inferior temporal lobe are impacted. So this makes stroke patients a valuable source of information and study in neuropsychology, as they're associated with very different types of symptoms and cognitive or perceptual problems that might arise from it in addition to motor problems. What is the cause of stroke? And the good news is that this is primarily a lifestyle disease and it is entirely preventable. Things such as the lack of exercise, poor diet, smoking, poor sleep, stress can all contribute to the occurrence of stroke. So it is never too late to take control of your lifestyle and make an effort to prevent or limit the possibility, the likelihood of stroke occurring, no matter the age. I hope at this point that all of these three diseases provide a clear illustration and a vivid one of what happens in the case of imperfect or faulty neural transmission and neural functioning. Okay, so now we should move into the other side of the action potential transmission. So by now you realize that we have discussed only half of the process of neuronal transmission. We discussed how the action potential is generated in the axon hillock, how it is conducted across the entire length of the axon, and how it reaches the axon terminal. However, perhaps one of the most fascinating topics in microanatomy is what happens at the synapse. How exactly is the signal transmitted to another neuron? And there are two basic mechanisms that can account for this and make it possible. The simpler one relies on electrical transmission. So what happens in that case is simply that the membrane of the two neurons that communicate with each other, the presynaptic and postsynaptic neuron, make contact and allow the passage of ions freely between the two cells. And the advantage of that is that the transmission of the signal is almost instantaneous. depolarization on at the end of one cell impacts right away the level of depolarization in the postsynaptic neuron. So this is especially useful if you're trying to mediate a process that should be really fast. For instance, one that generates reflexes, escape reflexes. And also it is quite useful when you're trying to synchronize the activity of many neurons with a high level of precision. However, it is also a rather rigid mechanism, not particularly flexible and not amenable to learning, right? And here comes into play chemical transmission, which is far more complex and it is much slower. Instead of being a process at unfolding within the time frame of milliseconds, it can take tens, hundreds of milliseconds, or even entire seconds and minutes. And that's a high cost. But the benefit it is that it is a much more flexible process. So whenever learning occurs within neural circuits, as a result of experience, let's say, then this type of transmission is one of the mediators of such learning. Okay, so why is it so complex? And perhaps one of the key players, if not the key player within the process of chemical transmission is the neurotransmitter. Okay, so what is the neurotransmitter? It is a molecule synthesized within the presynaptic neuron and stored within the axon terminal. Now, once the action potential reaches the axon terminal, what happens is depolarization at that specific location, and that triggers the opening of calcium channels, which further depolarize the axon terminal. But more importantly, this has an impact on the vesicles that contain the neurotransmitters and leads to this vesicles fusing with the membrane of the presynaptic neurons and to the release of the neurotransmitters within the synaptic cleft. It is not necessarily the case that only one type of neurotransmitter can be released through this process. The same neuron, the same presynaptic neuron can release multiple types of neurotransmitters, and that can also vary as a function of the fiery rate of the presynaptic neuron. So this is a fairly complex mechanism, as I already mentioned. Okay, so what happens next? Well, we have all these neurotransmitters released within the synaptic cleft, and what they do is bind with receptors on the membrane of the postsynaptic neuron, the spine on the dendrite. There are two basic receptors that can allow this process to happen. A simpler one, the so-called ligand-gated ion channel, and also the more complex G-protein coupled receptors. But we're not going to go too much into details regarding that distinction. Suffice it to say that both mechanisms, both types of receptors lead to the opening of ion channels within the synapse, within the postsynaptic neuron. And this can have both an excitatory or an inhibitory impact, depending on which receptors have been opened. And this is important to mention, that different neurotransmitters bind to different receptors. You can think about it as a key fitting into a lock. Each neurotransmitter is its own key, and it only fits into a designated lock. So depending on which lock you unlock, the result can be either an excitatory postsynaptic or an inhibitory postsynaptic potential. And you should keep in mind that this changes the membrane potential of the postsynaptic neuron, and we're only talking in this case about a single receiving neuron. But this postsynaptic neuron presumably receives information from thousands, tens of thousands, if not more neurons. And all of this information is aggregated both in inhibitory and excitatory at the level of the axon hillock, where the action potential might be generated as a result of this compound signal. Okay, so this is mediated fundamentally by neurotransmitters, and I already mentioned that different neurotransmitters have different properties. They bind to different receptors, and this is a fascinating field of study. There are more than 100 neurotransmitters that have been identified to date, and the impact is quite different depending on what part of the brain we're talking about, what type of neurons, and also what the conditions are for their release. A lot of this of the impact can be modulated by other neurotransmitters released at the same time within the synaptic cleft. However, for simplicity purposes and also in the interest of time, we're only going to talk about a couple of neurotransmitters which are both quite popular, both in science and more recently also in the media for a variety of reasons, and also specifically those that play a key role in some cognitive disorders and types of cognitive functioning. Perhaps one of the most popular neurotransmitters, largely because of its involvement in addiction and its popularity nowadays related to attempts to control addictive behavior, is dopamine. So what we know about dopamine is that it is produced it is synthesized within just a couple of brain regions, small brain regions within substantia nigra and ventral tegmental area, as well as the adrenal glands. However, dopamine is released throughout a large part of the brain through the dopaminergic system, and what that refers to is an entire system of neurons that are capable of synthesizing and releasing dopamine. So as you might expect, depending on what area of the brain dopamine is released into, the impact can be reflected in higher order cognition, in voluntary movement, or in reward or reinforcement learning. So different brain parts that release of dopamine impacts different types of cognition and behavior that we'll discuss in relationship with it. The other side of that impact is reflected in dysfunctional levels associated with Parkinson's disease, psychotic thoughts and delusional behavior, and addiction. And again, that is the logical outcome of the impact that dopamine has on reward and reinforcement learning. In this case, what happens is that abusive abusing that reward or reinforcement learning system leads to addictive behavior. And it doesn't necessarily matter what the source of the addiction is. It could be alcohol, it could be tobacco, it can be gambling, any type of object. It is ultimately mediated by the same neural mechanism. And the abnormal regulation of dopamine plays a key role in it, which makes dopamine especially interesting for the treatment of addiction, because if drugs can effectively and safely control the release of dopamine, that can mediate and that can help the control of this type of behavior. Next, norepinephrine. You can think of norepinephrine as the brain's fundamental mechanism of putting you into high gear. And this is hardly surprising given the location of its synthesis. The site of production within the brain is the locus coeruleus, within the pons, an area of the brainstem, which is specifically responding physiologically to stress. As a result of that, as you might expect, the levels of norepinephrine are quite low while you sleep, but they're really high when your body senses potential danger. As a result, the norepinephrine system is involved in the fight or flight response. And fundamentally what it does is generate arousal, alertness, vigilance, but also anxiety. It has very clear links to cognition and behavior. So, for instance, it is fundamentally related to attention. If you're vigilant, if you're alert, then your attention is much better. And also your ability to remember things might be much better. Mental flexibility is also one of the outcomes of engaging the norepinephrine system. If attention and memory are much better, unsurprisingly also you're much better, let's say, at finding different solution to a problem, thinking outside of the box, or switching tasks effectively. It is also linked to mood, and as a result of that, you might imagine that too low or too high levels of norepinephrine might not be so beneficial to the body. For instance, dysfunctional levels of norepinephrine can be associated, if too low, with Alzheimer's disease, with memory problems. Also, dysfunctional levels, too low or too high, can be associated with mood disorders such as bipolar disorders or depression. Norepinephrine, rather dysfunctional levels of it, are also associated with a fascinating condition called visuospatial neglect, in which people seem to be unaware of half of their visual world. And we'll have a more significant discussion of that topic later. Okay, serotonin is yet another popular neurotransmitter, and it bears a lot of similarity with norepinephrine, both in where it is generated, synthesized in the brain, and that would be again in the pons, in the brainstem, though in a slightly different location of the pons within the Raphe nucleus. But it is similar in the sense that it impacts a large network of cortical areas, as well as the cerebellum and the spinal cord. It has also select links, especially to mood, but also to more generally to cognition and behavior. For instance, it's got quite a bit of an impact on learning and memory, as well as on the motor system, on muscle contraction. As you might expect, dysfunctional levels of serotonin have been associated with mood disorders. So, for instance, levels that are too low are believed to lead to depression. And as a side note, some of the first antidepressants developed in the 60s were trying to modulate, rather to regulate serotonin, to up the levels of serotonin as a way to treat depression. More recently, serotonin has made the news, though, because of some very interesting findings. It's been pointed out that the majority of the serotonin production actually takes place not within the brain, not within the roughy nucleus, but interestingly enough, in the gut. So a group of researchers from Caltech estimated that about 90% of the serotonin in the human body is produced in the gut, and it is impacted by the healthy microbiome. So a healthy gut leads to good levels, normal levels of serotonin, while an unhealthy gut has quite a bit of an impact by lowering the levels of serotonin. And this relates to a much broader discussion relating brain functioning with gut functioning, to the extent to which some pretty wild hypotheses have been put out, some of which link traditional neurological disorders such as Parkinson's to activity within the gut. So whether disorders which are believed to have a neural origin ultimately reside in some other parts of the body, some of the gut, is something that remains to be seen and remains to see whether it is fully substantiated by empirical evidence. But suffice it to say that serotonin, amongst other components of neural transmission, has been a source of surprise to researchers in recent years. Okay, one last neurotransmitter that we will quickly go over is acetylcholine. And we know that it is synthesized within the basal forebrain, and that it impacts a variety of cortical areas, especially within the frontal lobe and the hippocampus. But not only, it also impacts activity within the occipital cortex, which is not clearly shown here in the picture. As a result, it's got select links to cognition and behavior. It has relevance for sensory processing and attention, for short-term memory and learning, but also it stands out by its role in motor control. This particular neurotransmitter can be found as an effective communicator in the neuromuscular junctions, enabling neurons to control muscles. And naturally, if the levels of acetylcholine are not optimal, let's say they are too low, one could experience weakness within the body. And in the case of pathologies, it can be related with certain conditions such as myasthenia gravis, condition related to dysfunctional functioning of within the peripheral nervous system that we'll have the opportunity to discuss later when we talk about motor control. Of course, since we might know that Alzheimer's disease has a damaging effect on the forebrain, it should come as no surprise that dysfunctional levels of acetylcholine can be encountered in the Alzheimer's disease. Okay, so with that being said, there is an interesting note to be made here, and that relates to the use dysfunctional levels or suboptimal levels of a specific neurotransmitter. And this is interesting because it provides an important challenge to psychopharmacology. And the main thing to notice here is that there is an optimal medium level for a specific neurotransmitter. So if that level is too low or too high, then performance or one aspect of performance, whether related to cognition or motor control, will suffer. Hence, we are trying to optimize. The body is naturally trying to regulate and optimize through its own internal mechanisms the level of neurotransmitter based on circumstances and our own internal state and what we're trying to accomplish. However, the optimal level is highly variable and idiosyncratic. There are vast individual differences in terms of what that level might be. Somebody's optimal level is not somebody else's optimal level for a neurotransmitter. Furthermore, the optimal level for a neurotransmitter, the level of a single individual, can be variable, and it is subject to modifications based on the impact that hormones have on neurotransmitter production. And also by the complex interactions that one neurotransmitter has with other ones. So that makes the challenge of psychopharmacology so difficult. Where does a person fall along the curve at a specific time? Hence the process of controlling the amount of drug consumption involves a process of prescription, observation, and refinement, specific to each individual. Okay, so at this stage we covered quite a bit of material and we are finished with our review of neurons and neurotransmission. So if you feel so inclined, you should feel free to take a break. Otherwise, we will continue our discussion next with fundamentals of the central nervous system. And we're gonna move to a completely different level of explanation and description. We're gonna move on to a macro level, which is in many ways more closely related to some of the things that we'll be discussing later during this term. Okay, then just to take a break from the brain, let's start our discussion with the human spinal cord. What the spinal cord does is effectively carry out information to the muscles from the brain with the aim of controlling their tone, regulating their activity, but also input signals from the body, from your skin, from your internal organs, from the muscles. So both input and outputs are fundamental in how we keep track of what's going on in our bodies and controlling our bodies. And the spinal cord hosts bundles of neurons and the axons organized into tracts to this end. Now, you are already familiar with the spine, the backbone, or to be more formal, the vertebral column. This is the bony protective encasement of the spinal cord, which is segmented into 33 different bones. There's a bit of an ambiguity about that in the book, so just to make it clear, there are 33 different bones or vertebrae, but 31 nerve roots. And now if you look at these pictures, the math might not quite pan out. You can see that we have 7 vertebrae responsible for the cervical division of the spinal cord. That would be your neck. 12 responsible for the thoracic spine, your rib cage, your mid back. 5 more responsible for your lumbar area, your lower back, then 5 fused vertebrae that constitute the sacrum and the part of the spine that connects to your hip bones. And sometimes we also add onto that the tailbone, the coccyx, which is in fact also made up of 4 much smaller fused bones. So I hope that by now that makes sense and our count is correct. So now, why do I mention this? Well, probably you're already familiar with major divisions of the spinal cord and of the vertebral column. If not, statistically speaking, most of us will encounter within our lives some back pain, and then those terms will very quickly come into play. However, more relevant is the fact that it is important if and where spinal cord damage might occur, and unfortunately that also happens quite a lot, and the results can be very debilitating, if not fatal. And one thing to keep in mind is that where it happens matters quite a bit. The higher it happens, the more debilitating its effects will be. And part of the reason for that is quite intuitive. At the higher part of the spinal cord, you have all this information that relates both in an input and output mode. Let's say the arms and the legs with the brain. But if you go further down the spine, then the nerves that connect the arms or the head with the brain have already left the spine. So you're only left with fewer body parts to control. Hence, if the damage occurs higher up within the cervical part of the spinal cord, the more debilitating and potentially fatal the results will be. If you want to intuitively keep track of that, you might remember the saying, C5 keeps you alive, or to be more precise, C3, 4, and 5 keep the diaphragm alive, which ultimately comes down to the fact that those 3 sections, these 3 vertebrae, host tracts that control breathing. So you can imagine if those tracts are damaged and breathing is impaired, then your life might be at risk. Okay, so then if we talk first about motor tracts, nerve bundles that connect the brain to your muscles, how are they organized? And there are several motor tracts. We're not gonna spend too much time talking about them. Suffice it to say that the major motor tract is the corticospinal one that starts in the motor cortex and then controls different parts of your body, and it stands out by the fact that it is responsible for voluntary movements. So whenever you decide to stand up, whenever you sit down, whenever you wave at someone, this corticospinal tract is involved in that process. We also call this a pyramidal tract, and that's for anatomical reasons, because it runs through the pyramidal portion of the brainstem. But what really matters here is the fact that it is one of the spinal tracts that we should be keeping intact at pretty much all costs. Now, as a point of brain organization, you might know that to a large extent, whatever side of the brain suffers damage, it impacts movement on the other side of the body, and that's because of the contralateral organization and communication of information within the brain and within the spine. So if you suffer damage from within your motor cortex, let's say within your left motor cortex, then you might have impaired motor abilities with your right hand side of your body. So for instance, typing things on your keyboard with your right hand. Okay, so this is the one of the major motor tracts, and the one that we pay particular attention to in the context of this course. However, I should briefly mention 4 other motor tracts just to give you a more thorough appreciation of how movement is controlled. Well, one is the rubrospinal tract, and it is, you can think about it as a backup tract when the spinal cord is damaged. Then we have the tectospinal tract that is responsible for the head and eye movements. We have the vestibulospinal tract, which as the name says, is responsible for balance. And last but not least, we have the reticulospinal tract, which is responsible for muscle tone and also for automatic locomotion and posture. So you might imagine that we cannot keep within awareness every aspect of body functioning and motor control. A lot of the moves, a lot of the movement that we are engaged in is completely automatic, and the reticulospinal tract is involved in this type of automatic movements. So to that extent, it complements the function of the corticospinal tract. Okay, so then let's move on to the somatosensory tracts, which this time carry information from the body to the brain. And 2 major tracts we are going to mention here are the medial lemniscal tract, which mediates proprioception and touch, along with the lateral spinothalamic tract, which hosts information about pain and temperature. And they're both obviously quite important. You might remember that proprioception is responsible for keeping track of where your body is in space, and we certainly have vision to help us with that. But proprioception complements visual information about your body position and state by carrying information from muscles. So for instance, knowing whether a muscle is contracted or extended can give you quite a bit of information about position. And this is obviously also important whenever visual information is missing. Let's say being able to keep track of the location and relative position of your limbs in space in a very dark room. Okay, this tract also houses the input that is coming from touch receptors. So it will let you know whether the touch, the intensity of the touch, and also the location of the touch, which body part has been touched and how strongly. Okay, the other tract is responsible for temperature and pain, right? Nociception. And one thing that you should realize right away is the fact that this organization allows the discriminate damage to one tract versus the other. So the possibility exists because of this type of organization. Let's say that the medial lemniscal tract is functioning quite okay, while the lateral spinothalamic tract is damaged, and hence you might not be able to receive information about pain and temperature. And this can happen and it is related with a number of diseases, also having a genetic component, those that involve atrophy of the pain receptors and the entire somatosensory tract that is responsible for those receptors. And while this might seem like a good thing at the beginning because you no longer have to experience pain, it is in fact resulting in a very debilitating condition. Pain is by and large how our body learns and how our brain learns. And it also critical for survival. If you don't know when and how your body should be in pain because it suffers damage, then this really puts your safety and your life in peril. Okay, so quite a bit of information in this picture, which also is featured in the textbook, but three things stand out here, and it would be a good thing to try and remember. One of them is the major organization of the spinal cord into dorsal columns and ventral columns. And simply put, what that means is that dorsal columns carry information from the body. It hosts somatosensory tracts, while the ventral columns take the information from the brain to the muscles through the motor tracts. The other thing that is quite obvious from this picture is the fact that gray matter, which contains the body cells, the somas, is at the center of the spinal column, while the white matter, which contains the axons of the neurons, surrounds the gray matter. And why is this interesting? Because this stands in contrast to brain organization, which features gray matter at the surface of the brain and white matter on the inside. And last but not least, and I hope this is quite obvious, all this information leaves or comes into the spinal cord and then to the brain through these spaces in between vertebrae via spinal nerves. This also has some very clear implication, or rather explanation, for why our spine is structured into so many segments. This breakdown into multiple segments not only is responsible for providing our spine with flexibility, but also allow the efficient transmission of information in and out of the spinal cord to the rest of the body. An immediate consequence of the way the spinal cord is organized is the dermatome architecture of the human body. So what is a dermatome? Well, a dermatome is a part of the body, especially a part of your skin, which is connected to the brain via a specific nerve, a specific bundle of neurons and their axons. And this has some interesting consequences for the diagnosis and treatment of various injuries and diseases. So, for instance, just to provide a quick example, while the brain has a good ability of detecting the source of pain, which part of the body is suffering in one way or another, the organization into dermatomes sometimes leads to some confusion as to which part of a specific dermatome is responsible for pain. So, for instance, because the same nerve connects the heart and the arms to the brain. If you're about to experience, let's say, a heart attack or other heart problems, they may be accompanied by left arm pain, which is surprising because the arm is not necessarily in any kind of pain, but there is a bit of confusion as to the specific location of the origin of pain within the framework of the same dermatome. Another point that we will revisit is about the location of potential injury of the spinal cord, whether higher or lower down the spine. And if the damage occurs at the top of the spine, let's say in a cervical area, then the damage can be quite extensive. It can lead in some cases to quadriplegia, a inability to control and sense all your four limbs. While if the damage occurs lower down the spine, in the thoracic region, let's say, then your arms might be spared, but you will suffer an inability to move, or at least severe weakness associated with control of your legs and of your feet in paraplegia. Okay, so this is a very consistent organization of the human body in terms of its connection to the brain, and as I mentioned, it's also got significant impact on how we diagnose different conditions. So, for instance, if your damage occurs to your lower back, which happens surprisingly often, it may lead to a tingling sensation in your legs, down your legs all the way to the top of your foot. So if the damage is lumbar, your doctor or health professional might try to diagnose the source of the damage by asking your experience, or questioning your experience of possible weakness or tingling within your legs and feet. Okay, so that concludes our discussion about the spinal cord for the time being. So at this time is probably a good time to move into our discussion about the brain. And all of you are probably familiar with this type of picture, this way of visualizing the brain from the outside. And this is relevant for mentioning a couple of key points regarding brain architecture. One is the fact that what we're looking at here is primarily cerebral cortex. This is what we see from the outside of the brain. It's the cerebral matter, the gray matter, that consists primarily in the bodies of the neurons. The other thing that we associate with brains is this wrinkly sheet. And this is because of the organization of the brains in what we call gyri or bumps versus sulci, the indentations within the brain. And in case a sulcus is deep enough, we have a specific name for that. We refer to that sulcus as a fissure. So this is how the brain looks from the outside. Gray matter with a wrinkly appearance due to the organization of the brain into gyri and sulci. And this is definitely one of the key pair of terms that you should remember, because most of the time when we talk about brain areas, we refer to those brain areas in terms of specific gyri or sulci. Okay, so what if we take a better look at the brain by examining a cross section? What a cross section of the human adult brain reveals right away is this organization into the cerebral gray matter on the outside, which is shown in a rather pink shade here, ironically enough, and the white matter on the inside. So why is this difference, at least in terms of color? And that has to do with what these areas represent. The gray matter contains the bodies of the neurons, the somas, as well as the dendrites, the part of the neuron that receives information, that accumulates signals. While the white matter contains glial cells and axons. The axons which are covered in myelin. And as you remember, myelin is a tissue, a fatty tissue, hence the white color of a huge aggregation of such axons together. Okay. Now, if we continue to look at the brain, and we are also starting to ask a bit more about the relevance of structure for function, at the most general level, if we try to take a bird's eye view, what we segment or structure the brain into are 4 different lobes, 4 different major parts. The frontal lobe, which hosts the motor cortex, responsible for movement. Areas that control speech production, such as Broca's area, and we had a conversation about that last week. And as a side note, it only makes sense that the motor cortex and Broca's area are within the same general area of the brain, since both of them are responsible for motor control. But also cognitive control systems. So what does that mean? Well, intuitively and rather informally, what that means is that the frontal lobe, to a large extent, is in charge of brain networks. So brain architecture and hierarchy is something that we'll have the opportunity to discuss in more detail in the coming weeks. But what that ultimately means is that some part of the brain needs to be in charge of what happens through these many cortical networks. And the frontal lobe hosts cortical areas that satisfy that function. So again, the name for that is cognitive control, and we'll have the opportunity to discuss that in more detail in the coming weeks. Okay, another major area of the brain is the parietal lobe, which accommodates or rather hosts the touch cortex. And also attention systems. Whenever we talk about attention, we will have to refer to the parietal lobe. Occipital lobe is responsible largely for vision, and it might be surprising that you have an entire part of the brain, an entire lobe dedicated to a single perceptual modality, vision. But vision is one of the key aspects and types of problems that your brain has to solve. So again, this is something that we will revisit in more detail soon enough. Last but not least, we have the temporal lobe, which we associate with audition, and this time that makes sense because it is right next to the ear, as well as speech comprehension. So you might remember our discussion about Wernicke's area, as well as memory systems. Okay, so 4 different lobes, 4 different types of oversimplified but still useful divisions in terms of cognitive function. However, this would hardly be fine-grained enough to be able to navigate a brain. So to that aim, each of the lobes is further divided into gyri and sulci. So for instance, if you look at the frontal lobe, you can refer to a specific area of it as the inferior frontal gyrus, then the middle frontal gyrus, and then the superior frontal gyrus, and so on and so forth. But there is a cost to that. So first of all, for many intents and purposes, not even that is sufficient to get the level of granularity that we need when I'm trying to talk about specific cognitive functions. But on the other end, sometimes that is comes at the cost of too large a level of granularity. And this relates to another idea. That is the idea of interindividual cortical variability. These are 4 different brains of 4 different individuals of the same age, the same IEQ, the same socioeconomic status, and so on and so forth. So after equating individuals across a whole number of variables which are presumably responsible for brain structure, we can still see a fair amount of variability in the size of different gyri and sulci. So just to make this clear, the overall organization of the brain stays the same. We still speak about the parietal lobe and the occipital lobe. We still talk about the same sulci and gyri, and the relative organization, again, stays roughly the same. But the precise size and geometric distances between these regions does change quite a bit from one individual to another. So now when we reflect about function and the consequence of interindividual cortical variability on function, what we should say is that it is still the case that across different individuals, let's say the frontal lobe is responsible for cognitive control and for movement. But if you're trying to zero in on specific parts of a gyri, of a gyrus or of a sulcus responsible for much more discrete tasks, let's say representing faces or representing visual inputs associated with reading, then the size of those areas and even their overall configuration geometrically in 3D is quite variable across individuals. So we should be very careful in jumping from structure to function. So if in one individual a particular area, let's say with a volume of a few millimeters, is responsible for one function, that doesn't necessarily mean that the structurally corresponding area in another individual satisfies precisely the same function. So just to summarize that point, by and large, we exhibit the same general anatomical structure, and we can systematically relate structure to function. But when it comes to explaining the relationship between structure and function, we have to pay a lot of attention to interindividual variability. Now, we have talked quite a bit already during this lecture and last week during the previous lecture about motor cortex and somatosensory cortex. And that's not just because they're so important for cognitive neuroscience, but also because they've served a guiding role in how we approach the study of other cortical areas. And they've provided some very interesting insights into the peculiarities of cortical organization. So, for instance, if you remember from last week, Wilfred Penfield championed the study of cortical organization by trying to figure out exactly how different information is stored within the motor cortex. And what he found was evidence for topographic correspondence between different cortical regions and the body surface. And this is a point that stands both for motor cortex and for the somatosensory cortex. What does that mean? So what he noticed is that if you stimulate a batch of neurons which are responsible for the thumb, let's say, then presumably the next batch of neurons is responsible for the fingers, and then the one next to them for the hand and the wrist, and so on and so forth. So this makes sense. This provides some very robust and meaningful and intuitive organization of the motor cortex and the somatosensory cortex. This organization is not entirely a match to what happens with respect to the human body. So for instance, if you take a look at the very end of the somatosensory cortex, you might note that the genitals are placed right under the toes, which anatomically is not correct. However, by and large, there is a correspondence between body anatomy and representation of these body parts on a body surface within this part of the cortex. Other thing that is not immediately clear is why some parts of the body are overrepresented. So for instance, you can see here that the face and the hand are overly represented. We have relatively large groups of neurons responsible for controlling these parts of the body and sensing what happens with these parts of the body relative to others, such as, let's say, the wrist or the elbow. And that certainly makes sense, because think about what a motor cortex does. It controls movement. So we definitely have good reason to have very fine-tuned and robust control of what we do with our hands and fingers. And we also need this type of control for our face and for our head, because this is primarily how we manage to communicate information with the outer world. So being able to express, for instance, emotions efficiently and effectively and correctly is highly important for social interactions, and it's something which is studied extensively both in social cognitive neuroscience and in vision neuroscience. Naturally, the same point stands for sensing, for the somatosensory cortex. To be able to control things well, we need to sense exactly what happens in that part of the body, be that the hand or the face. Now, one interesting source of confusion here for students who just begin the study of neuroscience is where this pieces of cortex are located. And if you look at this picture right here, it's quite clear that the motor cortex it resides within the frontal lobe, while the somatosensory cortex, which neighbors it, is actually in a different lobe. It is within the parietal lobe. One last point that I want to make about this is neuroplasticity. And that has to do with the fact that these maps are not set in stone. Okay, we got the overall topographic correspondence, and we have some magnification corresponding to some body parts, the face and the hand. But that doesn't necessarily mean that these areas cannot shrink or grow. So for instance, expertise, let's say perceptual expertise or motor expertise, can come into play to increase the number of cortical resources that come into play for controlling or for sensing. If you learn a musical instrument like piano or guitar, then it is quite likely that the piece of cortical the real estate involved in responsible for the movement of your hand and your fingers might get enlarged. To take another example, if one part of your body is atrophied, or if you lose it, let's say a finger, then what happens is a corresponding impact at the level of the motor cortex and somatosensory cortex. So that happens is that the neighboring cortical areas, which are responsible, let's say, for other fingers or your hand, might get a boost to take advantage of available resources in their neighborhood. So relatively steady, robust organization, but quite a bit of plasticity that can be induced either positively through training, whether moral or perceptual, but also through damage, injury, disease, and so on and so forth. Okay, now when we talk about somatosensory cortex, we're effectively talking about touch. It is touch cortex. But what about the other sensory modalities? Well, vision is very well represented. We have, as we already mentioned, an entire lobe, the occipital lobe, dedicated to it. And also there is quite a significant part of cortex also dedicated to audition, to auditory perception. But if you look at the rest of the brain, which is not marked here by blue or by purple, we see the label association cortex. So what does that mean? Well, it means that a large percentage of the cerebral cortex is actually not specifically dedicated to a sense or to motor control. Rather, what it does is integrate information from multiple sensory modalities and relates that with motor control as well. So this is where things start to become quite a bit more complicated. And the association cortex is also what subserves complex problem solving, decision making, language, production and comprehension. All of these high-level skills reside on top of many sensory and motor processes. So to be able to manipulate, to take into account all of this information and convert it into an output delivered to the motor cortex, we need the involvement of different areas of the association cortex. As you might notice here, for instance, a large majority of the frontal cortex is labeled as association cortex. And this is not really a surprise, right? We already know that the frontal cortex is involved in cognitive control, right? It is sitting, so to say, at the top of a hierarchy that is in control of what happens with the rest of the brain when it comes to cognition. It is also responsible for mood, it is responsible for decision making, and so on and so forth. This also makes the study of association cortex probably so much more difficult and challenging. And to add on top of that, since we already talked about inter-individual differences, if you're trying to evaluate the level of similarity across individuals, then that structural similarity and functional similarity is much more compelling for the primary and secondary motor cortex or somatosensory cortex than it is for the association cortex. While things get integrated, where things like language and memory and attention come into play, there is a lot more room in terms of flexibility for a specific individual on how his or her brain solves those problems. Okay. So now, just like we use cardinal points to orient ourselves in space, how do we navigate the human brain, right? So we have all these structures, we have all of these gyri and sulci, but sometimes we need a a broader way of referring the different parts of the brain and their relationship to each other, right? How do we say that the frontal lobe is placed in a relationship with the occipital lobe, for instance, in a 3D volume? And some interesting terms come into play here. And it's very useful to know what they mean. In some cases, it is rather obvious. In others, it is a bit less obvious, and these terms need to be learned, since we will come across them quite a lot. So if we try to navigate the brain as a 3D volume, one direction is superior versus inferior, or dorsal versus ventral. And what that means is that we're looking at the top of the brain or the bottom half of the brain. So for instance, we would use those terms to say that the parietal is superior to the temporal cortex. If we talk about the front or the back of the brain, we use the terms anterior or rostral and posterior or caudal. Whenever we talk about human anatomy, anterior and posterior are terms a lot more commonly often, while if we talk about animal brains, then terms like rostral and caudal come more often into play. So just to use them in one sentence, I would say that the frontal lobe is anterior to the occipital lobe. Okay, so these are two directions across which we can navigate the brain. What about the leftover direction, from side to side, from ear to ear? Well, that would be medial, towards the middle of the brain, versus lateral, towards the outside of the brain. Okay, but that is not enough. Whenever we look at brain pictures, such as those offered by an MRI machine, this is a structural scan of a brain of a human adult. We need to look at cross sections, right? We cannot visualize all the information within the entire brain at the same time. We perform sections throughout the brain, and we visualize this information as an image in a particular plane. So now we have different directions across which we can cut through to look at the brain. One of these directions is the sagittal one, from the front to the back, and this gives you information about what happens within the brain, both gray matter and white matter. In this case, you can also see, for instance, the cerebellum, and you can see the pons here, and so on and so forth, the corpus callosum. And this is quite useful, but whenever you look at this, you're losing quite a bit of information about what happens from the lateral to medial variation in brain structure. So then let's see how we can solve that problem. Another axis across which we can cut through the brain is an axial one, and this is by and large the most common one, because it enables you to look at both hemispheres at the same time. You can see what's on the left and you can see what's on the right. However, if you do that, you lose a lot of information about what happens from the superior to the inferior navigation through the human brain. So then, what other direction can we conceive of to visualize brain structure? Well, the remaining one is the coronal structure, which cuts through the brain from ear to ear, essentially, and you can see superior and inferior brain structures at the loss of the anterior to posterior direction. Typically, if we look at brain images within a scanner, we try to evaluate information in all three planes, because that gives us a much richer way of visualizing and appreciating information regarding a cortical area, let's say, by looking at it in the context of all three different planes of view. So these are terms that you should become familiar with, because they're especially relevant whenever we look at brain pictures with the use of MRI. Now, the MRI has become invaluable both for clinical and for research purposes when studying gray matter. So, for instance, if you're trying to find an insult to the gray matter, or if you're trying to visualize cortical activity using functional magnetic resonance imaging, then MRI is really valuable. Nowadays, with the advent of new techniques that provide better spatial resolutions, we can visualize what happens to thin strips of gray matter, lamina by lamina, which is pretty impressive, because different sheets of neuron within the gray matter portion of the brain can subserve different functions. And we had a conversation about that last week. Some parts of the gray matter, some sheets of neurons are responsible for receiving information, some for integrating, and some for sending. So MRI is making all of that possible. However, in the older days, the MRI was not particularly useful for informing us about white matter. And that also reflected a certain prejudice that if you care about cognition, then perhaps it's not as important to study the white matter, the axons. Ultimately, you're interested in what happens at the level of the neuron and the dendrites, and that should be sufficient. However, that prejudice and that mistake has been corrected, and with the advent of techniques such as diffusion tensor imaging, we have been more recently in a position to visualize better and better what happens with fiber tracts, with bundles of axons that connect different brain regions with each other. And this is definitely important for research and for clinical purposes. For research purposes, it allows us to better understand what parts of the brain communicate with each other. So a more robust bundle tell us that some parts of the brain have privileged connectivity with each other, and they can influence each other much more strongly. And that certainly has implications for how cognition is framed and how it is structured. What are the building structural blocks that make it happen? But it also has high relevance for clinical purposes. For instance, in a lot of conditions, let's say those associated with consistent trauma to the brain, concussions, structural MRI might not be able to tell us anything about insults to gray matter, and we might feel safe to conclude that there's been no damage to the brain. Yet, in some of those cases, DTI has been efficient in showing that there has been damage to these tracts. Their integrity has been compromised. And though the cerebral cortex might be intact, it's the ability of different cortical areas to communicate with each other effectively has been also compromised. And this can have many consequences overall for somebody's health, but also for cognition. So the MRI is a versatile machine nowadays that can allow things as different as taking pictures of gray matter through and analyze it with the use of structural MRI procedures, analyze cortical activity as related to different functions and tasks with the use of functional magnetic resonance imaging, but also visualize the anatomy of white matter and provide some very compelling pictures of this true highways of the brain, as illustrated here. Okay, now we have discussed a bit the cerebral cortex and the white matter, but what about some of the subcortical structures? And we will have the chance to discuss some of them in more detail in the coming lectures, but I'm just going to mention a couple of them and their primary function as we know about it at this time. So for instance, one of the main systems associated with emotion processing is the limbic system, and the amygdala is one subcortical area that often comes into play. It is an almond-shaped area of the brain, hence its name, amygdala. That tends to react especially strongly is as elicited by negative emotions such as fear or disgust. We have already talked about the corpus callosum. It is the bundle of white matter fibers, axons, that connect the two hemispheres of the brain. And we have also mentioned the importance of the temporal lobe for memory. And one area in particular in that sense is the hippocampus, as well as the entorhinal cortex. They're specifically the object of study for memory researchers. And not just for memory. As a side note, more recently it's been found that these parts of the human brain are also important for integrating perceptual, visual information with memory. And that's definitely something that I'm looking forward to finding out more about in the next couple of years. One other areas that I will mention is the thalamus. And that is because it serves as a relay of information, especially for motor and sensory information. So before the visual information, for instance, reaches the occipital cortex, it passes through the thalamus. And the same is true for most senses and for motor information. However, more recently, the thalamus has become a lot more popular, both in science circles and in the media, because of its ability to influence vigilance and awareness. Right, so it became a key area, a hot topic of discussion when it comes to consciousness, for instance. Anatomically, in the proximity of the thalamus, we can also find the basal ganglia. As a matter of fact, the basal ganglia tends to wrap laterally around the thalamus on both sides of the brain. And it is one of those regions of the brain which plays a key role in the initiation of movement. Hence, no surprise that in Parkinson's, where this area is damaged, the result is impaired motor control. Also, it has an important role in learning new motor skills, whether that is learning how to swim or learning how to wield an instrument or a bat. So again, a key area within motor control and learning new motor skills. Now, there are a lot more names and brain structures represented on this slide, but this is just to provide an anatomical context. It is not something that you necessarily need to study in detail or will be questioned during the exam. The midbrain is again a highly complex part of brain structure, and we're not going to have time to go into much detail about it today or for the rest of the course. But I'm only going to single out a couple of its substructures, which are labeled here with a red asterisk. I would note the periaqueductal gray matter. which is responsible for gating pain. And this relates to one of the previous points, and the usefulness of pain for learning purposes and for keeping track of the state of the body. However, it is beneficial in certain circumstances to block pain, for instance, if that is essential for survival. If you have to be able to move efficiently in a adverse environment, and pain is distracting you from that objective, from survival, then the periaqueductal gray matter plays a key role in blocking temporarily pain and its distracting effect on behavior. So again, just a temporary fix, but an adaptive, valuable use of neural resources in the service of survival. The substantia nigra is another interesting part of the brain, and that's one of its key roles is to synthesize dopamine and send it to the basal ganglia, so you can initiate movement. And it is one of those areas which is impacted by Parkinson's, and as a result, it has a very negative impact on the ability to control movement. Last, I would also mention the crus cerebri and the red nucleus, which are essentially paths for motor neurons, descending motor tracts. The cerebellum is yet another one of those brain structures that have been misunderstood for many, many years. It has been correctly believed that the cerebellum is involved in motor control, in motor coordination. If the cerebellum is damaged, you might experience difficulty with motor control or with speech. It is responsible for controlling the vocal tract, the tongue, the mouth, and so on and so forth. Also, if the cerebellum normal activation and processing is impaired, let's say because of alcohol consumption, this is also one of the expected outcomes. Difficulty coordinating movement precisely, and also slurred speech. However, the cerebellum is extremely densely populated with neurons, so it almost seems like an abuse of resources. Why would you need such a heavily populated brain structure if all it does is just control movement or fine-tune movement? Not that that is not a complicated problem, but many scientists thought for the longest time that this is an evolutionarily old structure, a vestige of an older brain structure that fulfilled some other functions. In reality, in more recent years, a lot of cognitive neuroscientists have revealed that the cerebellum is active as viewed, let's say, in fMRI scans, whenever we are involved in language processing, memory processing, and even perception. To be completely honest, it's been one of my sources of surprise the first time I conducted an fMRI scan related to face processing, to see activity, robust and systematic activity in the cerebellum. So at this time, there is more or less agreement upon the idea that the cerebellum is involved in cognition, memory, language, perception, but how and to what end, that remains to be seen. It remains to be researched, and it remains to be clarified. Okay, one last thing that I want to mention before we conclude today is the importance of protecting the brain. If the brain is so fundamental for so many different functions, then naturally the body created quite a few different mechanisms for providing safety for the brain. And probably the one that we're all familiar with, because it's quite obvious, is the skull. It's a relatively thick, sturdy, bony layer protecting the brain from the outside world. However, if you perform a craniotomy, if you cut through the skull and remove the skull, what you see right away is not the brain, but instead what you have access to is the meninges. And that refers to three different protective layers that encase the brain. The dura mater, or leathery like very robust surface underneath which we find two other finer surfaces, the arachnoid matter and pia matter, as well as cerebrospinal fluid that fills the gap between the arachnoid matter and the pia matter. So again, this is protecting different layers and types of protection for the human brain. However, this system also has its own downside. I already mentioned that brain swelling is quite dangerous and potentially fatal. However, the same applies to inflammation of the meninges, and that's exactly what happens in meningitis, right? And the problem with that is if a part of the brain or a part of the meninges swells, then that puts pressure on a certain part of the brain, because there is limited physical space volume within within the human skull. There is no space for the brain to expand into when pressure is applied. That can result in a lot of problems. It can result in a temporary dysfunction of a cortical area, or it can result in neuronal death. So an entire part of the brain might suffer damage because of that. So by and large, the human biology has provided us with a complex and versatile way of protecting of the brain, but this also has its own limitations that we should be aware of. And with that being said, we are concluding the discussion of brain anatomy today. We will round this off and add a few additional notes next week. However, most of the time will be spent next week moving into a different and equally fascinating topic, that is methodology in cognitive neuroscience research.